Okay. Yeah. Hi, we're on. Okay. So if there, what was I saying? Yeah. So yes. So where are the external hydrogen bonds, and how many are there? Sorry.
Depends on the residues.
Oh, wait. Just for the backbone. Let's say they're all alanines. That make it easy. They're all alanines. Usually alanines will make a helix, by the way, but let's say these are very special alanines. They want to be in a battery. Yes. So how many external hydrogen bonds are these two chains going to make? Any? Are there any? Yes. There will be some external hydrogen bonds. Where can we find them? Let's go along the chain, right? So here we have the nitrogen, which has a satisfied internal hydrogen bond, right? Now if we go here we have carbonyl, which has a satisfied hydrogen bond the other way around. This nitrogen also wants to make a hydrogen bond, but it has to go up. Right? So this is, if this is the whole protein, then this will be what? What? Water? Yes, water. Yeah. So it will make a hydrogen bond with water. Okay. There should be oxygen somewhere here, which isn't drawn. Not here. This is. Where's the other one? This is another nitrogen, so there should be an oxygen somewhere in here, pointing up. Not drawn. I don't know why. This is the next. Oh, yeah. This is the next nitrogen. Going up. Should make a hydrogen bond. This is the same. But there's missing an oxygen here again. Right. So there's 1, 2, 3 nitrogens sticking up, and there should also be 3 oxygens sticking up, which aren't drawn in this figure. Yes, I'm never going to use this figure again. I'm sorry for that. The one that I hand drew, which is in chapter 1, that's better because we checked it, and because you've been looking at it and you didn't find any flaws. So it's a double check. Okay, so there's 6 unsatisfied hydrogen bonds at the top, and another 6 at the bottom. Right? Okay, so that's external hydrogen bonds. So why is it so important to have these internal hydrogen bonds satisfied? So now we know the internal ones, right? So okay, let's get back to that question. So what are internal hydrogen bonds? Of the protein, right? So with itself, yeah? Okay. That could also be with the side chain, but it has, yeah? But they're inside the protein. What happens if you fail to make one of those? Well, you could, but water wants to make 4 hydrogen bonds. So if there's one, let's say there's one backbone amide, NH, that wants to make a hydrogen bond, you put a water there, then it wants to make 3 more hydrogen bonds. So if there are 3 more hydrogen bonding partners over there, then the carbonyl would probably be able to do that by itself already. So that's not a likely thing. So the most likely thing is that it will just be there by itself. And does anybody happen, maybe somebody with a chemistry background, happen to know the amount of energy that's typically associated with forming a hydrogen bond? It's about 2 kilojoules per mole, which is about half a kT. A kT is a thermal energy, and at room temperature. So that means, and the stability of a protein, we've already told you, it's not very stable. It's stable enough, but for a protein to be functional, it should be stable, but it should also be, you should also be able to disable it and to clear it away, right? How to say that? To degrade it. Yes. If it's too stable, you can't degrade it. Yeah. So if you have a particular protein that you generate in your cell that's too stable to be degraded, then you'll end up with only that protein in the end. Yeah? So that doesn't work. So proteins are only marginally stable, and their stability is on the order of 2 hydrogen bonds. the energy associated with two hydrogen bonds. So that means if you have one unsatisfied hydrogen bond in the core of your protein, you've already halved the stability of your protein. You have to deduct that from your. If you fail to make two in different locations, obviously, because there could be two amide hydrogens. That would be the worst case when they're like two, because they can't do it. It has to be an amide with an oxygen, right? Or a hydroxyl with a carbonyl. Yeah, then you've lost complete stability of your protein. So two missed hydrogen bonds is enough to destabilize a whole protein. So that's why it doesn't happen, because there's energetically no room to make that amount of mistakes. Okay, so now, right. So have I gone through this? Yes. Protein folding, fold prediction, function prediction, and alignment. If you have two particularly distant homologs, where the sequence identity is very slim, what would be the first thing you use if you want to align to very distant homologs? You should know this. Even if you haven't done ASA, what's the trick to find a good template for homology modeling if you can't find close homologs? You want to find a distant homolog. Side lots. I wouldn't use side lots. It's better than blast, but there's something much better.
Hidden market models?
Yes, so what do they have in common, side lots and hidden market models? They use profiles, right? So if you're looking for a particularly distant homolog, what would you do? Sequence profile? Profile, right? So that's one way to do it. Another way to enrich your sensitivity for finding distant homologs is to make use of secondary structure information, because that's typically conserved, right? So if you're using already at the level that you're using profiles, you can actually use the profile to take secondary structure. We'll get to that in a minute. And then you can look at the aligned secondary structure to see whether the matches are good. Okay. And you can even use it directly in. So there's Praline, the Jaap's alignment program. It has a mode where it actually predicts secondary structure for each of the input sequences, and then favors alignment of same predicted secondary structure elements while doing the pairwise alignment, and that seems to happen as well. Okay, so the other thing is, so this is why would it be useful to have this prediction, but the course up to now has been about structures and about also deriving secondary structure from three-dimensional structure. So why would you need to predict it if you can have the three-dimensional structure? Well, I hope I don't have to explain to you that we have many, many more sequences that we don't have a structure for. So there's many more sequences in the database than structure. So for that gap, this phrase is getting a bit old, but it's just I don't know any other way to do it. Mind the gap, right? Okay. And please notice that these bars are the same, right? You only have experimental secondary structure for a protein if you have the three-dimensional structure, because that's the only way to do it. You can get some sort of very rough measurements of the fraction of helix and sheet in a protein from NMR or circular dichirism, but they don't tell you where it is, only how much you have of it. So if you're looking at a specific residue, you can't tell you that this residue has to be here. Okay. Well, this one we've already done, right? So you've been working with DSSP, or at least the output of it, and you might have seen these letters in the output files of the DSSP, right? Where you have the eight-state secondary structure assignment, which has obviously the helix and the beta strand. Then you have the beta bridge, which is sort of the opposite of an extended strand. It's two hydrogen bonded residues from different strands, but they're the only ones. So it's like a single residue beta sheet, or two residues, one on each side, right? And since people didn't want to call that a beta sheet, they call it a beta bridge. Then there's two other flavors of the helix. You can wind it up tighter, then it's a 310. Or you can wind it up looser, then it's a pi helix. Yeah? They do occur in structures if you look for them, but they're a minor fraction. So if you're doing secondary structure prediction, typically people just lump these in with helix. And then there's two types of turns. There's the type 1 hydrogen bonded turn and the type 2, which is a band. But these are typically looped in with coil. But these are, so if you're doing secondary structure prediction, these actually look a lot like the twist that you make in the backbone for alpha helix. So they're in 5 psi combinations, they're very similar to alpha helix. But they're sort of like a single or only two residues of alpha helix, which can't be helix. But it could be a tight turn for a beta sheet. So you have a beta sheet coming up, then a two residue turn. The two residue turn in a beta helix, beta sheet, sorry, is typically this one. Okay, that's already way too much detail. So DSSP is not the only one. There's a couple of other ones, and there will be two more on later slides. Most of this work has been done somewhere in the end of the last century, so quite a while ago. DSSP is still the most widely used because everybody uses it, everybody keeps using it because you never have to explain why you use it because everybody uses it. This is the way you write papers, right? If you can choose between 3 methods and you don't really have a strong reason to pick the exotic one, you just pick the one that everybody has because then no reviewer can complain about it. That's what everybody uses. And then this is what I will explain. So there's 3 flavors of helix, which we then lump into helix, and so on. Okay. It does matter which one of these 3 you use, and I'll show you that on the next slide. So if you look for DSSP, the amount of helix, strand, and coil, it has about 50% of coil. And then you take the 2 other ones, stride and define, you see they actually identify a little bit more helix, but mostly more strand at the expense of coil. Obviously, it has to add up to 100%. So if you predict more of something, then you predict less of something else. So there's about 10% of residues that DSSP will call coil, which define would say, but it's actually strand. Yeah? And this is surprising that the biggest sort of confusion could be between strand and coil. You can imagine if you have a long beta sheet, then somewhere at the end maybe it starts to get a bit floppy or irregular, right? And then it depends on your precise definitions of what you call a hydrogen bond or which 5 psi combinations you still allow. If you say, it really stops here and this is something else. Or you say, well, it's still parallel and it's still hydrogen bonding. Maybe it misses one hydrogen bond, but then it continues. And it has different 5 psi angles here, right? And then you can extend your beta sheet for a few more residues. Yeah, so that's what, and that also happens with helices, but much less so. Because the helix is already quite regular by itself. There isn't that much you can change in a helix before it really you really have to break convincingly one of the hydrogen bonds. Okay, so to make this graphical, this is just one sort of random example. Here's the PDB code if you want to reproduce this for yourself at some point. So this is this is not the whole protein. We're only showing this is a beta barrel protein, and we're showing only the beta sheet assigned residues here. So this is from the top, where you can see that it's sort of the barrel, and this is from the side. But you see the barrel is sort of, it's incomplete. It doesn't have all the sheets. So there's a gap here. If you then take, this is yet another one, PALC. This is from 2005. You see it predicts another strand here, or predicts, it assigns another strand here. This is not prediction yet, right? This is still going from a PDB, a three-dimensional structure, to similar structure assign. It finds another strand, and apparently this is two, you can see that there's a twist in here. Right? So apparently DSSP then says, oh well, this can't be a strand. And then if you take B spider, which is also from 2005, it finds another smaller strand, and then you have the complete beta barrel. And now you can see that it really goes all the way around. Have you seen beta barrels before? Yeah. So the nice thing about a beta barrel, so the funny thing about a beta sheet is that it has an edge. At some point, you stop, and then if you're not at the end of the protein, you have a problem, because you have all these unsatisfied hydrogen bonds at the edge strand. The beta barrel solves this problem by just looking around. Yeah? Okay, and they're very common for enzymes, because it's very easy to make very specialized binding sites inside this cavity. But they're most dominant as membrane pores or channels. Because you can imagine it's very easy to put hydrophobic residues along the outside of the barrel, so that they're stable in the hydrophobic interior of the membrane, with some polar residues at the edges, so it anchors in the polar head group region of the membrane. And then the inside you can coat with whatever you want to make a certain molecules go through. Okay, and you can make them any shape, any size, because you can just add strands or remove strands and make it bigger or smaller. Okay. Propensities, Sanna already covered this, so I can be very quick about this. You've seen this particular slide, I think, even probably. So if you look at the propensity for a particular amino acids to be in helix or strand, you find different. You find very different patterns. And the order of the residues is not the same between these two slides, these two panels. So here alanine is at the top, because it's the most likely one to be found in a helix, relatively speaking. Yeah? Because alanine is also the most dominant residue. It's the most often occurring one. So that you find a lot of them in the helix is not surprising. But you find a lot more alanines in helices than you find alanines elsewhere. If you look up alanine in a beta sheet, it's over here. It's below 0.8. Yeah, it's a big difference. Okay, I don't want to go through all the other details. So now we're going into secondary structure prediction. And I'm just wondering whether this is a good moment for a break. Shall we do that? Yeah? So let's be back at 5 to 10, and over coffee think about. So this method just uses the propensities and some very simple rules, like how do you start a helix, how do you end a helix, and what are the tricks you could think of to do better than this? This is the oldest method in the book. It's 1974. Yes, I was not even playing with Lego at that time. Or maybe with Duplo. I don't know. But this is there's a lot of improvements being made since then, and let's see how much of those you can come up with. Yeah. Okay. See you back in 10.